Before DNA can be replicated, the double-stranded molecule must be unzipped into two single strands. In order to unravel DNA, these bonds and the base pairs must break. The unraveling is accomplished by an enzyme known as DNA helicase. During DNA replication, the double-stranded DNA molecule separates, and each strand is used as a template for the construction of a new strand. This is the result in the formation of two identical copies of the original double-stranded molecule. This is called semi-constitutive replication. The helicase disrupts the hydrogen bonding between the base pairs and separates the strand into a Y shape known as the replication fork. This is where replication begins. DNA is directional in both strands, represented by 5 and 3 and a 3 end. This means each side group is attached to the DNA backbone. The 5 end has a phosphate P group attached, while the 3 end has a hydroxyl OH group attached. Once the DNA strands are separated, a short piece of RNA called primer binds to the 3 end of the strand. The primer always binds at the starting point of replication. However, the replication fork is bidirectional, which means one strand is oriented to 3 to 5 direction, the leading strand, while the other is 5 to 3, the lagging strand. The two sides are replicated with two different processes to support the directional difference. Because replication is formulated in the 3 to 5 direction on the leading strand, the newly formed strand is continuous. The lagging strand starts replication by binding various primers. Each primer is only several bases apart. Then the DNA polymers can add pieces of DNA called Okazaki fragments to the strand between primers. Once elongation of the DNA strands are complete, the strands are checked for errors, repairs are made, and the sequences are added to the ends of the DNA. Replication now produces two DNA molecules, each one strand from the parent molecule and one new strand.
Transcription is the first step in the protein synthesis, but the second step of the central dogma. It is a process in which RNA is synthesized using a DNA template strand. As the DNA enters the RNA polymerase from 3' to 5', it is splitting into two strands. The DNA parent strand is paired with RNA base pairs, A with U instead of T because RNA uses uracil instead, and C with G. mRNA job is to carry genetic information that is copied from the DNA in form of sequences. RNA polymerase is an enzyme that is responsible for copying a DNA sequence into an RNA sequence. As the DNA is moving through the RNA polymerase, RNA gets longer until the DNA strand gets to its end. Transcription factors help the RNA get synthesized and copied correctly. You can see in the beginning of the start codon how there are extra nucleotides on the mRNA sequences. This allows a barrier around the protein. Translation then begins, which is the process of translating the sequences of mRNA molecule to a sequence of amino acids. As seen, the mRNA strand is attached to the small ribosomal subunit. Translation begins with the ribosome along with tRNA carrying the met start codon binds to mRNA at the promoter, AUG. This is when translation begins, and it is known as the initiator complex. tRNA anticodons takes place in the site of A, P, and E. tRNA moves into the P site, which holds the amino acid chain. After another tRNA goes into the site carrying amino acids, the ribosome starts matching the tRNA anticodons with its complementary codon to form an amino acid chain. In the A site, the amino acid is attached to one another by polypeptide bonds. And in the E site, it pushes out tRNA out the ribosome and it adds another new one at the A site. The tRNA anticodons are paired with their promoters. It reaches a stop codon at the UAG and it's called a release factor, which ends the process.